Have you ever just stopped to think about the sheer miracle of sight? I mean, really think about it. Every sunrise, every face you recognize, every single word you read, it all starts inside this tiny paper-thin piece of tissue at the very, very back of your eye. Today, we are diving deep into the retina, the incredible biological machine that literally turns light into your reality.
Okay, so let's get our bearings. Think of your eye kind of like a camera. Light comes in through the lens at the front, and at the very back you've got this, the retina. You can think of it as a biological movie screen, where the world is first projected. It's actually an upside-down, two-dimensional image of everything you're looking at. But here's the thing. This is no ordinary movie screen. It's a supercomputer. So that brings us to the biggest question of all. How does this tiny, almost transparent membrane take simple light and kick off the process of building your entire visual world? Well, let's find out.
So, to answer that big question, our first stop is to really nail down the retina's most basic role, acting as that movie screen for the eye.
You know, the official definition calls the retina a light-receptive, innermost nervous layer. But the most important part of that whole phrase is nervous layer. The retina isn't just eye tissue, it's literally brain tissue. It's an extension of your central nervous system that just happens to be located in your eye, and it's got the incredible job of converting light into the language of the brain, and that language is neural signals.
Now that we know what it is, let's take a little tour of its surface. If you were to look directly at it, you'd see it has some pretty important landmarks, almost like a geographical map.
Looking at the back of the eye, we see two absolutely critical landmarks. The first is the macula, that darkish spot right in the center. That is your high-definition camera. And the second is the optic disc, that bright circle where all the wiring bundles together to leave the eye. So the macula is all about sharp, detailed, central vision. It's the part you're using right now to focus on these words. And right at its heart is the fovea, which is just packed with special cells for the absolute sharpest focus possible. The optic disc, on the other hand, is like Grand Central Station. It's the big exit point for all that visual information heading off to the brain. But here's a fascinating consequence of that design. Since the optic disc is just a big bundle of nerve cables, it has zero room for the cells that actually detect light, so it can't see at all. Every single one of us has a physiological blind spot in each eye, a tiny patch of the world that you are physically incapable of seeing. And the reason you never notice it, your brain just brilliantly fills in the gap for you.
Want to find yours? You can actually do it right now with this slide. It's a super simple but pretty amazing experiment. Okay, try this with me. Go ahead and close your right eye and focus your left eye only on the X. Now slowly move your head closer to the screen and then back away. At a very specific distance, that O on the right will completely vanish. That's it. You've just found the exact spot where the optic nerve leaves your retina. Pretty incredible, right? All right, so that was the tour of the surface. But the real magic, happens when we zoom in. Because the retina isn't just a single layer. It's an incredibly powerful 10-layer neural computer. And we're about to look inside. This is a cross-section of the retina. Just look at that intricate, layered structure. Now, what's really interesting here is that arrow. It's showing the direction light travels when it enters the eye, and it reveals something completely backwards from what you'd expect. Get this, the light-detecting cells are at the very back of the retina. This means a photon of light has to travel through all the other layers of neural wiring, all the processing circuits and output cables, before it ever hits a sensor. It's like putting the microphone at the back of the speaker. And while it seems like a bizarre design, it actually puts these high-energy cells right next to their vital blood supply, which keeps them nourished. It works flawlessly. So, to simplify those 10 complex layers, you can really think of them in 3 functional groups. At the back, you have the light-sensing layer, the photoreceptors that catch the light. In the middle, a processing layer of interneurons refines that signal, and at the front, the output layer of ganglion cells sends the final packaged signal off to the brain. So let's zoom in even further, right to that first light-sensing layer. It's time to meet the stars of the show, the photoreceptors themselves. Okay, so we have 2 types of these light-catching cells, rods and cones. And the numbers are just wild. We have a staggering 120 million rods, which are incredibly sensitive and give us our black and white night vision. Then we have far fewer cones, only about 6 to 8 million. But they are the absolute powerhouses that give us our sharp, detailed color vision in bright light. And this graph shows their layout perfectly. See that huge green spike right in the center? That's the fovea, and it is jam-packed with cones for that high-def vision. The purple line shows the rods, which totally dominate our peripheral vision and peak in a ring around the center. That makes them perfect for detecting motion out of the corner of your eye and seeing in the dark.
Now, our ability to see in color comes from 3 different types of cones. We have short wavelength, or blue cones, medium wavelength, or green cones, and long wavelength, or red cones. By comparing the signals from just these 3 cell types, your brain can perceive the entire incredible spectrum of color.
All right, now for the climax of the whole story. We've seen the structures, we've met the cells, but how does a particle of light actually become an electrical signal that the brain can understand?
Well, this process is called phototransduction, and it is truly one of the most incredible molecular events in all of biology, and it's happening trillions of times a day right inside your eyes.
Here is another one of those mind-bending facts. In complete darkness, your photoreceptor cells are constantly firing away, releasing neurotransmitters. They're on. It's the arrival of a photon of light that actually shuts them off. So the signal for light is really the stopping of the dark signal. So strange, but true.
Here's a simplified look at how it works. On the left, in the dark, these tiny channels in the cell are wide open, letting a constant stream of ions flow in. Think of it like a running faucet. This dark current keeps the cell on. But on the right, when a single particle of light hits a special molecule, it triggers a chain reaction that instantly slams that channel shut. It turns the faucet off. The flow stops, the cell goes quiet, and that sudden silence is the signal for light. That is the birth of vision.
You know, when early scientists like the brilliant Santiago Ramon y Cajal first drew these intricate cells, they used the word retina, which is Greek for net, because they imagined it was just a simple net for catching light.
But we now know it is so much more than that. It is not a passive net. It is an active, pre-processing computer. It sharpens edges, detects motion, and adjusts for light levels long before the signal ever even leaves your eye. It is truly one of the most powerful and efficient neural computers on the planet.
So the crucial takeaway is this. Your retina is where the raw signal of light is meticulously built piece by piece. But it's your brain that takes that signal and builds the rich, 3-dimensional story of the world around you. Which leaves us with a final question to think about. What really is the difference between simply seeing and truly perceiving?